Okay, good afternoon, everyone. Thank you for joining us for today's webinar. And my name is Cassie, Marketing Supervisor at GenScript, and I'll be the moderator for the webinar today. So a little background about GenScript for those who are new to us. GenScript is the leading life science research and application service and product provider in various fields, from basic life sciences research to translational biomedical development, industrial synthetic products, and cell therapeutic solutions. The topic for today's webinar is cPass neutralizing antibody detection for COVID-19 post-vaccination monitoring. So let me quickly introduce you our speaker for today, Dr. Yanfeng Li. So Dr. Li is the Head of Business Development for IVD Division in GenScript Asia Pacific. She's well-trained in cellular biology and immunology with extensive research experience in signaling pathways, especially in B-cell biology and T-cell functionality. She authored in more than 10 world-renowned journals and led more than 50 successful clinical trials, connecting insights from consumer behavior, skin biology, and intervention efficacy. This webinar will take around 45 minutes, and we'll have about 15 minutes for Q&A session later. You can type in your questions in the chat box, and we'll answer them after the presentation. If we didn't get to answer your questions during the Q&A session, we'll email back the answers to you after this webinar. So without further ado, we'll let Dr. Li to start her presentation right now. So Dr. Li, over to you.
Thank you so much, Cassie. Good afternoon, everybody. It's really my pleasure to have everybody joining today's session. I know I actually personally invited many of my friends and colleagues to listen to this topic. So if there's any questions, feel free to ask them during the session or connect with me afterwards. As Cassie has just mentioned, today we're going to talk about cPass neutralizing antibody detection for COVID-19 vaccination monitoring. So basically, this cPass technology has been a wonderful success among the three parties, Duke and US, A*DXD, and also GenScript. So I will walk you through the agenda of this talk. So basically, I'll try to extensively introduce about the SVNT technology. SVNT stands for the surrogate virus neutralization test. So I oftentimes getting people's questions, right, from various backgrounds. So basically, now that we are in the vaccine era, well, everybody will ask, what are the vaccine efficacy? The actually different vaccine platforms offering the same efficacy between, especially the popular ones from China and also the ones from US, the inactivated vaccines versus the mRNA vaccines. And everybody actually get the same level of protection post-vaccination. In terms of immunity longevity, right, the antibodies or protective immunity will be the same for all invaders. Vaccine evaluation, so basically there are so many different technologies out there. What are the key differences and between the traditional antibody detection method and also our SVNT technology? In terms of the variants of concerns, this is in the news every day. So basically, everybody's concerns are like, how can I cope with the new variants of concerns? Now that Delta is actually, everywhere and, taking over the infection, actually, individuals, numbers are growing every day. So these are the questions that many of us are actually wondering. And I'll try to cover this and basically hope that by the end of this webinar, you're walking home back, knowing better the pandemic and also various of resources that are available for us to cope and exit from the pandemic, situations. Of course, lastly, I will talk about our cPass IVD kits for you to find from the resources available for us to get cPass tests. So these are the current landscape of available technologies for COVID-19 detection. As you can see on the left, is mostly used extensively from last year is to capture the acute infection stages to determine if someone is actually infected by detecting the presence of the viral material. So these technologies include the qRT-PCR or the rapid antigen test. However, after the acute infection stage, right, these viral materials will be difficult to catch. And what we can do in the post-infection or post-vaccination stages are to look for antibodies. So in terms of antibody, there are two major pillars. Number one would be the IgG, IgM test that most of us are very familiar. These are called binding antibodies. So they can use swab or blood testing to look for the binding antibody response to detect the IgG and or IgM or others that's full sets of antibodies to actually determine the immune response to the virus. So mainly this is to detect the infection history and has anybody actually being exposed to the virus. What is more important is also the functionality of the immune response. So that has been traditionally defined by the virus neutralization test. And for VNT is to measure the neutralizing antibodies, right? So basically, this is to detect the neutralizing antibodies that can block the virus to infect the cells in our body. So I talk a little bit more about the implications and significance about measuring neutralizing antibodies later on. First, let me introduce the innovation of our SVNT technology. On the left, you can see that the virus can actually invade our human cells. So basically, the virus RBD, the receptor binding domain of the spike protein, can attach to the SCE2 receptor, and that interaction will allow the virus to be internalized and to replicate in our body. So in the presence of any neutralizing antibodies, you will be able to see that this interaction between RBD and SCE2 will be prohibited, and the virus will no longer be able to invade us. So by this principle, the SVNT technology was actually introduced. Basically, this is a blocking ELISA methodology. So by adopting the key principle of this viral-human interaction, RBD proteins and SCE2 proteins were in vitro expressed, and you can see that SCE2 receptor can be coated on the ELISA plate, and also the RBDs are actually conjugated to HRP to be able to induce a color changes during in the presence of or the absence of any neutralizing antibodies. So basically, no neutralizing antibodies and all these RBDs will be binding to SCE2 receptor, giving a strong signal, color signal. And in the presence of neutralizing antibodies, the RBD will be washed away. So basically, you'll see a very clear unchanged color. In this way, we can determine the presence of how much of a neutralizing antibody, in our sample. So let me introduce about, what is the significance of measuring neutralizing antibodies. By right, basically you can see from FDA guidelines of development and licensure of vaccines to prevent COVID-19, it actually detects that the three arms of immunity will be measured in any clinical trials. So that includes, especially during the vaccine, development work, right? So basically, T cell cellular response, and humoral response binding antibodies. Last, most importantly, is also the functional immune response measured by neutralizing antibodies. So neutralizing antibodies are not only important during the vaccine efficacy evaluation stages, but of course it's also important for each one of us, even after the vaccine trials have been completed, to still understand the individual differences, because it's apparent that not everyone will respond to a vaccine the same way. So basically, in terms of the presence of percentage of protection a vaccine can bring to each one of us, there are definitely differences. Well, someone will have a high immune response and others will not. So basically, that's why even after all the vaccines are approved for general population use, it's still very critical to us to understand our immune response against the vaccine that we are receiving. So secondly, in terms of the convalescent plasma treatment, I think in 2020, convalescent plasma treatment was actually quite popular due to the lack of any treatment, right? So there is this investigational COVID-19 convalescent plasma by FDA guideline, and indicates that measuring the neutralizing antibody titers is critical to determine who will be eligible for donating their convalescent plasma samples. And you can see that at least 1 in 160 titer of neutralizing antibodies is actually recommended to identify those individuals who are having a high titer. So this 1 in 160 is actually a good titer that can save other people's life. And for us who are getting vaccinated, even though there's no official way of telling us what is a protection level, we do wanted to see our own immune response to also have this level of neutralizing antibodies. So last but not least is, the various of treatments available for COVID-19. And you can see that FDA from very early on, November last year, has already approved a number of treatments that are composed of neutralizing antibodies as COVID-19 treatments. So these various of aspects does indicate that the neutralizing antibodies are playing a great significant role in us fighting against COVID-19 and getting that protection level. So basically, as you can see that for convalescent plasma treatment, even though the live virus assay it was set as a threshold of 1 in 160 and above, so GenScript product, our cPass technology has been validated to be equivalent by when you see this inhibition rate is larger than 68%, it detects that it's offering the same, results as having a live virus assay of giving a 1 in 160 titer. So basically, what is the difference, right, between the live virus assay and our new technology? As you can see on the left, it's not easy to run a live virus neutralization test. You have to go, set up a biosafety level 3 laboratory. Individuals, operators working in this lab have to be extensively trained, and even by entering the lab, poses you a significant hazards, right? So basically, you have to hood up and wearing the various of PPEs, protective equipments. The experiment itself takes multiple days to perform. Requires a very high level expertise to read even the results. Expensive, not scalable, and of course, because we're using multiple biologics, right? The virus itself is live, and the cells are live to culture. So basically, due to all these differences, it's going to be not comparable between lab due to the protocol or material, inconsistencies, right? Basically, every lab will have their own stock of the virus culture and the cell cultures. So this new technology, our SVNT technology, actually brings a groundbreaking, innovation to replace the troublesome, gold standard live virus assay. So currently, instead of having the whole virus, we have already purified RBD as a protein and also SCE2 without having to grow your cell monolayers. We have SCE2 coated on our ELISA plate. And this assay can be used in any BSL-2 labs. The requirement of BSL-2 lab is not due to our kit. It's actually to protect our operators who are running the test, so that we'll still treat each of the blood sample as contagious. So basically, one will still be preferred to actually run this experiment in the content hood. This experiment is much faster, 1 hour. Of course, it's automatable. Many ELISA platforms actually can adapt this assay. So the cPass test can actually be done at high throughput. As the results, as later on I can also share with you, actually much more consistent. We can actually follow our standard protocol so that the global harmonization can be easily achieved with minimal intervention. So that generally, I think the bring home message is that SVNT is actually creating a new paradigm for neutralizing antibody detection. And now that with this new technology, having a functional antibody test is not bound to BSL level 3 labs. Well, many of the countries even cannot afford to set up a lab, right? Basically, it's extensive maintenance work, even you set up one lab up. So basically, often people will actually ask, right, if FDA actually says that neutralizing antibodies are actually so important in the various of guidelines. Can neutralizing antibody testing predict protective immunity? Right. So basically, we quoted a few of stakeholders here. John Hopkins, actually renowned, entity, and Dr. Fauci. So basically, everybody actually tend to agree that the neutralizing antibodies are actually the gold standard for protective immunity. Of course, we're not trying to argue that T cells are not playing a role, but in a way that we needed to measure it extensively in a large population. T cells are just not possible to have a standard IVD method to allow that kind of scale. So we don't want to only, quote these famous people, but also we wanted to show you the data, right, as we are all scientists. So basically, this paper has been published last year, and it was actually an early accomplishment of this group in Seattle. So what happened is there was a fishing vessel. You went on the sea before everybody's departure. All the individuals were actually sampled for the RT-PCR results to look for the presence of viral materials, and luckily all the samples were also obtained or for serology analysis before and post their departure. So basically, everybody, of course, have to be negative in order to board this boat. And 6 of them are interestingly already, infected before this trip. So there are, 4 of them are actually having a higher IgG index, and 2 of them are slightly lower. So cPass was, SVNT cPass was used in this study as well. And we can see that 3 of them actually had high blockage, and 3 of them, even though they have the presence of binding antibodies, their neutralizing antibodies were not present. And you can see, right, while there was an outbreak, unfortunately, 85% of attack rate, basically among these 6 individuals, were wondering, right, what would be the best biomarker to indicate the level of protection. So I think very apparently, right, this is 100% agreement for those 3 individuals who had positive, cPass SVNT technology readings. They are all protected against this reinfection. And those 3 individuals who also had positive binding IgG antibodies, but lack of neutralizing antibodies, these 3 individuals are unfortunately all infected again. So basically, the conclusion of this paper is that the presence of neutralizing antibodies from a prior infection was significantly associated with protection against reinfection. So I think that's why it gives us a very good reason by measuring our neutralizing antibodies level after, no matter whether we have been exposed, reinfected, or we have received our vaccination, so that to understand whether our protective immunity can be achieved after the immune response. The second paper, which has been very extensively cited recently, is actually published in Nature Medicine. So basically, this paper actually is a very smart way of doing science. This is a paper published by Australian scientists. So basically, what they have done is to consolidate all these 7 vaccine trials. The x-axis actually data from the phase 2 clinical trials is to look at their neutralizing titers. The y-axis was the protective efficacy that has been reported, in the phase 3 clinical trials. So as you can see, there is a very significant association between the neutralization titer on the x-axis and also the protective efficacy in the y-axis. So this is very significant, right? Basically, among all the data that has been, revealed in the prior experiments, the data mining work actually indicated that most significantly association is actually between the neutralization titer and also the protective efficacy. So as you can see, the various of technologies, for example, Moderna, Novavax, and Pfizer, they are actually at the top of neutralizing antibody generation and also reported the highest protective efficacy. On the last was actually, the inactivated vaccine from the Sinovac team. As you can see, the level of neutralization antibodies are actually below that was detected in the convalescent patients. In this way, right, basically, conclusion was to show that neutralization level is highly predictive of immune protection. So basically, I think that gives us a very good reason to really identify how much of neutralization antibodies are we obtaining after our vaccination programs. And there's also, of course, a side news is that by looking at the decay of the antibodies, the trial authors actually also found that decay of neutralizing titers in even the vaccinated subjects are actually as rapid as the decay of that is observed in convalescent subjects. This means that our vaccine is not going to protect us forever, while we will have to monitor our neutralizing antibody levels along this journey to indicate how much of a protection I may have, and decide later on. As you can see, many of the manufacturers started to indicate that you may need a booster, a yearly booster or half yearly booster, depending on how much of neutralizing antibodies we may still have. Right, so basically, among these various of vaccines that have been reported here, you can see that there are various of technologies. Of course, the viral vector, RNA. These are the major, components from Moderna, Pfizer, and of course, 3 of them actually using the viral vector and technology. So all these 3 methods are actually relatively newer and newer vaccine approaches. On the right, while you see the protein technology as reported from the Novavax, and also cured or attenuated virus that is being adopted by the Sinovac technology. So various of different vaccine platforms, they are using different technology to generate the vaccine formulations. And of course, we're seeing different efficacy, as we just seen in the last page. And also, if you look more carefully, a array of different adverse events that was reported. So I think, to be fair, while we're seeing less of efficacy that came reported from the Sinovac vaccine, we also were actually delighted to see that the attenuated virus itself is actually a more proven and being tested technology, which actually was also reporting slightly less adverse events during the trial, and for people who are getting the vaccine jabs. So to be honest, I'm actually also waiting for my vaccine jab of Sinovac this Saturday, and it will be a very exciting journey to just look at the level of antibodies during the course of the vaccine regimen, and also follow up after a few months. Of course, I actually have the luxury of participating in this study to look at my antibody levels every month, right, to see the decay. And when that third booster is actually available, definitely I wanted to sign up for it when the antibody levels are not potent enough. So I think, as per also reported from the various of manufacturers, you can see that now that we are actually coping with the variants of concerns. Of course, on the left, you can see a list of those highly contagious and also highly concerned variants. Alpha is actually first detected in UK, Beta from South Africa. Of course, Gamma was reported first in Brazil, and now I think most concerning is the Delta, right? The B1617.2. Most recently, second wave infection we have observed in India. So no matter what, right, we'll actually still see the various of vaccine companies are still trying to report that the current vaccine technologies are still offering quite potent protection against these new variants of concerns. Of course, new research are still being undergone to evaluate, what are the new technologies that we can, evaluate to see whether there are any better technologies that will be able to, offer various spectrum of protection against even the future variants of concerns. But I think, as per the current technology and the manufacturers reporting, many of the current vaccines are actually offering good neutralization titer levels, above the levels that are expected to be protective. So in this way, I think it's a very positive signal that as we have already this many options to choose, I think we needed to do really is, when the vaccines are coming to our availability, we'll just need to take it up immediately. After you have received your vaccine jab, it will be important for you to understand your own neutralizing antibody responses and how long our antibody can actually last. So in this study, we actually have followed 200 individuals. Of course, by the 9th month, we still had a total of 164. And you can see that not only, not everybody is having the same immune response. Some are actually negative if, even after they were hospitalized, they didn't have any neutralizing antibodies. Those individuals who had positive neutralizing antibodies also had different waning profiles. So you can see in the center, actually 90% of the cohort, while 30% of them actually are rapidly waning. You can see that by 3 months of time, half of these individuals already didn't have, measurable neutralizing antibodies. And this rapid waning group, very sad, you see nobody actually had neutralizing antibodies after 6 months. So for the slow waning group, I think people are still more exciting, but some of them does, lose their neutralizing antibodies by 6 months. And as we are speaking, right, the floor friends and colleagues may wanted to debate and also, to hope that we're all sitting in this group, which is called the persistent group. So this group actually had long-lasting neutralizing antibodies, and we have done a very comprehensive, interesting machine learning algorithm to indicate that, this level, but also previous data from SARS, right, which is 17 years ago, that these individuals sitting in the persistent group, they may have their neutralizing antibodies available up to decades. Right? So this is a very good example of data set for us to, to individualize, personalize our vaccine regimen, especially for our third booster, to see, when you may need it, right? So basically, people who are sitting in this group may need it even 3 months after their first, full regimen. People sitting in the persistent group, they will have their antibodies available after decades, so there's no need for us to actually go, experience the pains and, potential adverse events, but we may already have sufficient antibodies for our protection. So basically, the international unit is something that, have received a lot of attention. WHO has came out with this international standard. So basically, these are common standards that are essential to test between different platforms and also eventually offer as the gold standard for companies, to actually work out their assets and, protocols. So as you can see, in the case of hepatitis B, the protective level of the antibodies, right, is actually 10 IU per liter. So I think for Hep B, people will have to go through this examination to enter certain careers. For example, laboratory researchers, you needed to see that you have that antibody to protect you from any potential exposure. So you can see on the right is actually our newly, formulated international standard for anti-SARS-CoV-2 immunoglobulin. And we actually offer 2 units. Basically, the international unit is actually being reserved for measuring the concentration of neutralizing antibodies. So when our cPass, SVNT technology has finalized our evaluation of, the kit versus this international standard, we'll be able to report international unit per ml. And this will be a very significant step because eventually WHO and many other organizations will start to accumulate data to allow us to understand what is the level of protection, right? Basically, the IU per ml is a significant step, and which we are actually, offer a lot of extensive resources to try to get the authority approval to allow cPass to report in IU. Of course, all the other antibody tests, brought in by the major diagnostic companies, they are actually using, major the traditional technologies to measure the binding antibodies, for example, IgG, IgM, etc. So in that, calibration against the international standard, the binding antibody assays will be able to report in the binding antibody unit. So that will be differentiating between, when we are testing ourselves with the right technology for neutralizing antibodies, and we'll be able to know that the international unit for protection and whether where we are sitting, whether it's above that protection or below that protection, is going to be a very critical piece of information. So the international standard is actually produced last year, and WHO has done a very beautiful summarizing of this study. A total of 45 labs across many different countries actually participated in this study. Of course, while you can see that while PRNT, the live virus assay, even in this summary has shown quite a bit of deviation between the labs. Our cPass was also part of the submission, and two of the labs actually use cPass, and you can see cPass was able to give much more consistent readings for the samples that WHO has sent across to these labs, in a blended way. And even the two labs themselves, they didn't know which are the other labs that are actually using this test. So our SVNT technology actually demonstrates very good between lab consistency, and of course the data validated by WHO group and the labs that are participating in this study are actually very beautiful. So our SVNT is actually currently used by many of the our vaccine trials and studies. Of course, I would like to show more, but of course, also some other studies are still under confidential information. And here I just talk about a few studies that have already been published. So on the left, you can see, right, that individuals who are post their infection can have variants of levels of neutralizing antibodies. And this is the timeline that was measured in the baseline. When these individuals actually received their vaccine, and their antibodies were measured again 3 weeks after the regimen, only 1 dose, right? So individuals who are naive to COVID actually have elicited a spectrum of different antibodies manifestation. However, for those individuals who recovered from COVID, was actually found to have formed a very high level of neutralizing antibodies even after 1 dose of the jab. So this data actually indicates that for individuals who may have already recovered from a prior COVID episode, it may be good for you to actually look at the antibody level post 1 jab of vaccine, instead of going through the standard protocol of 2 immunizations. Because as I said, immunization actually, you eventually have to look at your immune response in the individual level. And once the antibodies are actually hitting high enough, there's no need for us to go through that additional booster, because antibodies are a sweet spot, right? You can't have too much. Of course, you needed to have sufficient to actually offer that protection. Data on the right also shows, right, basically when studies measuring the binding antibodies, including IgM, IgA, IgG, and also in the meantime measuring the cPass neutralizing antibodies, the level, the level of seroconversion that we can see between the binding antibodies are very much higher. As you can see, at least 10 of the individuals reported a seroconversion after the first dosage. But for neutralizing antibody, you can see only 4 out of 20 was shown to be positive. So in a way, this is another data set showing that, to measure binding antibodies is not sufficient. And, to understand the neutralization antibody, is best to actually, getting a SVNT technology cPass test, because that is a direct measurement of neutralizing antibodies and a best correlate of protection. So as I think people are still worried about the variants of concerns, we are also subjecting our technology to see whether the SVNT technology will be able to differentiate neutralization antibody neutralization to the variants of mutants. Here, we actually had a study from Singapore. And you can see that 3 individuals, right? So basically one is actually 52 subjects, subject number 52 and 34 and 46, right? The highest this for 52 was actually experienced infection by the original virus. And when SVNT technology was used to evaluate the variants of neutralization against the mutants, you can see that people who recovered from infection against the variant strain were wild type strain will actually receive the highest neutralization for the wild type virus. And of course, the level of neutralization against UK is still very high. However, this individual, when you ask whether this person will be protected against the South Africa variant, the answer unfortunately is actually no, right? So neutralization against the South Africa variant is basically unpresent. Second individual who were actually infected by the UK strain, as you can see that the neutralization against the UK is actually quite robust, but against the South Africa strain is very much lower. And the last one is actually an individual who were exposed to the South Africa strain. And of course, this person had the highest neutralization against the South Africa, virus. But the level of protection against the UK and wild type was actually reduced. So we had done similar studies in US, and of course, using 4 different types of sample. One is a negative sample without having the neutralizing antibodies. Everybody did not manifest any neutralization against the different variants of concerns, of course. The sample that had weak positive protection, weak positive neutralizing antibody, actually that was shown that the wild type had the highest neutralization and subsequently reduced neutralization against the other mutants. The sample that was in the category of moderate positive sample, again having the highest neutralization inhibition to the wild type strain, but very much reduced to the rest of the mutants. So the last one, even the strong positive sample. So basically, whenever you look at your cPass results, which is currently still the wild type strain, you will expect yourself to have somewhat reduced efficacy against the other mutants. And this is the data that we have accumulated from individuals who have received their vaccines. Study from the US again. We had a few individuals receiving different platforms of vaccines, Pfizer, Moderna, Johnson & Johnson, etc. And you can see that the Pfizer vaccine actually showing the highest protection against the wild type variant. Of course, those inhibition were actually somewhat reduced to the different mutants, for example, South Africa and UK. And if you will look at this Johnson & Johnson vaccine, while it was able to offer a relatively acceptable neutralization against the wild type strain, the neutralization against the South Africa is very much negative, and even the UK mutant, the Johnson & Johnson vaccine was actually not fairly offer good neutralization ability. So this data indicates, right, I think in the era of the pandemic, while it may not be ending soon, we might have to live with it. It becomes endemic, and also it's very good. It will be helpful for us to keep monitoring our immune response and ensure that we will have the sufficient protection against the different mutants, especially we're still lacking the data. Whenever we have that delta mutant capacity neutralization available, we'll be happy to share with you as soon as possible. On the right is a study that was done in Singapore. Of course, serum samples were actually collected from the wild type and the UK and South Africa neutralization assay. Very similar to the previous results. When we received Pfizer vaccine, majority of us actually had enough neutralization against the wild type and also the UK mutant, but there's a significant reduction of protection against South Africa variant. So the data, I think the Delta mutant is the next data to come, but we'll expect it to be somewhat similar to the South Africa mutant. So if one have to ask me, right, what is the difference between the binding and neutralizing antibody assays? We'll have to say that, not only they are measuring different classes of antibodies, but the binding antibody assays might not be able to differentiate between the various of mutants. So by a few of the mutations that we observe in the RBD domains or the various of other locations, when you look at the RBD binding of the different individuals, while we see a significant differentiation between the neutralization capacity. However, if you are just measuring the binding antibody test, that differences is not able to be captured. So how do you differentiate neutralization to the variants of concerns? So this is a way for us to adapt our cPass technology. We can actually, other in the same time, in the same, sample, we can get that neutralization level for both against the original strain and also the variants of mutants if you can add the other RBD HRP variants to actually allow that in parallel analysis for our samples. So another very important feature, right, by adopting SVNT technology is that we are able to offer now the finger prick method to allow people not only, have to subject to venous blood sampling. So we have collected various of samples in Singapore, and that's constitutes of positive and negative samples, and of course in various of neutralization capacity. And we found that the agreement between, finger prick serum and also venous sample are actually 100% positive and negative agreement. The correlation was found to be really, beautiful, right? Basically, you can see the data is aligned very well on this line. And now that, you're not needed to go through a painful venous blood sampling, by pricking the fingertip, you'll be able to, draw enough sample for our cPass test. So people often ask, right, if my antibodies will decline, how can I, understand who and, when do I need my booster shot? So I think Sinopharm executive and also Pfizer CEO already declared that a third COVID vaccine dose will be likely needed within 12 months. So basically, these are information that generally the general population may needed to go have another jab, after 12 months. But I think in terms of public policy, it's easy to say. But in terms of our individual management, in eventually, as I just showed, right, everybody have different level of neutralizing antibodies at the start, after your immediate vaccine response. And these antibodies may actually likely, when at different speeds, right? So we may not want to just follow the guidelines, but because we're already seeing many cases of breakthrough infection, while we don't know how much of antibody we may have in our blood, right? Just blindly, having the same regimen for everybody after, a booster dose at 12 months after your original regimen may not be the best outcome to protect each one of us. So I think oftentimes our vendors, our collaborators ask, there are so many different assays out there. Why is GenScript SVNT technology most suitable for post-vaccine monitoring? So as I wanted to share that the SVNT technology still remains the only FDA approved kit actually to determine the functional neutralizing antibodies, and is recently been granted patent. So basically, the USPTO office have recognized that our new innovation is meaningful to offer additional benefits to the general population. As we have already talked, cPass results eventually will be able to report in the IU. Of course, this is a feature that we're still working very hard on, and we have been used in multiple vaccine studies. Of course, many of the international laboratories actually independently validated our test kit, and we have received many approvals around the globe for authority registration. So the potential applications of cPass SVNT technology will be first to identify vaccine efficacy, to look at your immune response and whether you have generated enough antibodies for your protection, to actually evaluate whether somebody may need neutralization, may need a vaccine. So if you were receive a cPass test even before vaccine, if you have been exposed to the SARS-CoV-2, that would be a benefit for individuals who may actually experience adverse events even for the vaccine jab, right? So when the time comes, the booster screening will be another important application, so that we can identify who and when we may need this booster shot. And apparently the SVNT is already being very much helpful for WHO and other world organizations to trace the origin of SARS-CoV-2 and to actually forecast and prevent any potential next outbreak. So last, I think I'm going to close my talk by actually introducing our cPass IVD in vitro diagnostic kit. So we have actually received first in Singapore HSA PA approval, provisional authorization for COVID-19 detection last year. So basically that allow us to use cPass to detect presence of infection, and also whether your vaccination is actually working, to determine who has the neutralizing antibodies. The data that we have submitted to the various authorities are actually beautiful. Well, we have compared positive and negative samples and found the agreement to be 100% for negative and also 100% for positive samples. And if no matter where you are, so I will recommend if you have your phone here, you can actually scan this QR code and get in your cPass booking through our dedicated service partner, Licensure. And cPass test is also available in many other countries, UAE, India, Philippines, thanks to many of our hardworking pioneers who actually recognize the importance of this new assay and wanted to offer this test to individuals who may need it to end this pandemic. Of course, if you are actually from other regions, cPass is also available broadly in US and also in Europe. And with that, I would like to thank you for all your attention, and I'll be happy to address some of your questions. Thank you so much.
Thank you, Dr. Li, for the great presentation. We have quite a number of questions from the floor, so let's go through it one by one. Okay, the first question, are neutralizing IgG antibodies and binding antibodies are IgM?
Cassie, this is I think a great question from the audience. So for neutralizing antibodies, right, it may or may not be a single isotype of IgGs. So I would say that for individuals who are sampling their blood at a longer time point, for example 3 months after vaccine or after infection, you're likely to see more of IgG as neutralizing antibodies in your serum. However, if you're looking at other sample types, right, for example you're looking at a saliva sample, that there was a paper showing that the neutralizing antibodies can be largely IgAs as well. Binding antibodies, is actually the all the antibodies that we call after we are exposed to virus or bacteria. Antibodies are the adaptive immune response when we actually encounter any of the pathogens, right? So binding antibody is the totality of antibodies, and neutralizing antibody is a subgroup of binding antibodies. But we are just, to look at this functionality of those antibodies who can block the virus and human interaction. So I think back to the question, binding antibodies is not only IgM. Binding antibodies contains all the isotopes of IgG, IgA, IgMs, and whichever you can call, IgG, IgD even, right? So basically, I think that is probably a basic immunology question. And I would like to, explain more if we have the luxury of, have a connection post this conference.
Okay, thanks, Dr. Li. The next question from the floor. After vaccination, frontliners are not checked at all for their neutralizing antibody levels. How low will the neutralizing antibody levels will be before they will need to get another booster jab?
Yeah, so this is a great question. I think we're trying to work very hard, in partnership with many organizations around the globe to ensure that the frontliners can receive in time their testing for neutralizing antibody levels. In terms of how low the neutralizing antibodies levels before you getting a booster, as we can see, right, if we're using the current cPass, it means that at least I will say that we'll be more comfortable when our neutralizing antibodies are above 68%. That is while you're having enough power to save another person's life, right? But if your antibodies are actually positive, that is above 30%, which gives you quite a good confidence that you still have the presence of neutralizing antibodies. Of course, ideally, if we were to give a threshold, I think to the wild type strain, we wanted to see us having at least 50% of neutralizing antibodies as per the current technology goals. So whether we have a booster available or not, that can be another question. But I think we actually see many MOH organizations actually started to discuss about, offering that booster, especially we're seeing so many cases of breakthrough infections in Singapore and also in the other parts of the world. So I think eventually this will be a political and scientific question at the same time. I think we will be happy to work with the local authorizations to actually determine what are the levels for a booster. But I think as many of us don't really have that long-lasting neutralizing antibodies, it will be very critical to first understand what is the level and bring that to the doctor to actually have a consultation to see, when should I receive my booster. This is a great question. Thank you, V10.
Okay. Thanks, Dr. Li. Another question from the same attendee. Can I get tested for neutralizing antibody against Delta and Delta Plus variants?
I think as of today, the original wild type, the Wuhan strain, is available as an IVD product. In order for us to receive neutralizing antibody detection against the Delta or the Delta Plus, currently, I think we'll be happy to talk with Tingwei to actually start, see whether you'll be happy to enroll as, our study, so that I think currently the additional attributes of protection against the mutants are still considered for research. We'll definitely like to, work this out when there are sufficient, interest from the community to have such evaluation against the Delta and, the Delta Plus. As of today, it's not IVD approved yet, but we'll work hard against that, goal.
Okay, thanks, Dr. Li. The next question, is it possible to use the cPass to titrate immune serum to obtain a neutralizing titer? Is 20% or 30% cut off a better threshold?
Can I actually have the first half of the question again? Cassie, thank you.
Oh, okay, can, no problem. Is it possible to use the cPass to titrate immune serum to obtain a neutralizing titer?
to titrate the serum, right? Basically, I think this probably is already our cPass user question. The cPass is currently, not by using direct from the sample. We will titrate down the serum 1 in 120 in the current protocol, right? Basically to allow us to detect a larger range. Of course, we can always do a serial dilution, just exactly as what the live virus or the pseudovirus assays are, using to understand the IC50, which means that the 50% of inhibition power while at certain titration. So yes, to the question, we can titrate the samples down for various of reasons for sure.
Okay, thanks, Dr. Li. The next question. There are some other brands of kits available commercially. How do you differentiate these kits since they are also detecting neutralizing antibodies?
Great question. So for as I just shared, right, basically, we are the first one. We actually GenScript work closely with Duke NUS, the inventor of this technology, to actually commercialize this product. And I think the neutralizing antibody is a very old concept, but in terms of commercial products, this is the first time, right? Basically, so we're very proud to be the first working with our very renowned scientist, Prof. Wanlin-Fa, who invented this technology. And I think in terms of differentiation, definitely we have already accumulated, more than 1 year plus experience by using this technology and keep improving the features. We understand this is a popular concept, so many companies also follow suit and also develop their own version of neutralizing antibody test. To be honest, I believe cPass is always the best because we actually working with the best scientists. We keep optimizing our features of the product, and so far we actually have being granted by the patent. So if you actually this question come from a diagnostic lab, I believe for diagnostic laboratories is the very much the first priority for us to guarantee that we're using a technology that is legally offered while there's no disruption in our product lines, because I think by having a kit from any of the unauthorized producers, it actually poses a lot of concerns in terms of the quality of the results and also the type of I think that to be one word is that I think GenScript is still by far the original of the kit, and we definitely offer the best possible support to your testing experience, and I think probably the gold standard of neutralization. Even Roche, they're trying to compare their binding antibody assays versus cPass, right, to claim that they are detecting neutralizing antibodies. So in a way that I think we are proud to already created so much of attractions from the audience, and I believe there's more to come and there's a larger application to it.
Okay, thanks, Dr. Li. Okay, one more question from the floor. It's very interesting to see a correlation between cPass inhibition of 68% and VNT 1:160. Can I ask, is this PRNT or CPE, IC50 or IC100?
Yeah, so the 1 in 160, I think we have actually submitted our data to FDA. The group actually manages convalescent treatments. So the cPass was done just by the original protocol. The comparator method is actually PRNT, plaque reduction neutralization test, which is the gold standard as per FDA recommendation. So we used PRNT 50 to identify 1 in 320 titer. So 68% equals to a PRNT neutralization titer of 1 in 320. Yeah, so that's when we actually offer a very good signal that, when your sample is above 68%, you're actually having a very good high titer neutralization.
Okay, thanks, Dr. Li. Maybe we can take on with this one last question before we end the session. Will the treatment of the sample prior to using cPass affect the results? For example, chemical inactivation of sample versus heat inactivation of samples.
That's a great question. I believe diagnostic labs, when we set up the test, we do want it to have minimal impact to operators, right? So that our operators who are working on these samples, which are potentially contagious, protected. So we needed to protect our operators and the team. So I think chemical treatments or heat inactivation are common practices being used in the industry. For cPass, I will recommend to go with heat inactivation because in that sense, you actually, introducing minimal intervention to, the solutions of the testing. So we have done a comparison between the non-heat inactivated sample and samples that have been heat inactivated. The results are very much comparable.
Okay, thanks, Dr. Li. So due to time constraint, we'll end the Q&A session over here. So for those questions that we didn't get to answer just now, I'll email the answers from Dr. Li back to you. So thank you once again, Dr. Li, for the great presentation, and to all of you who joined us today. And I hope you find this session today beneficial to your work. So thank you and have a nice day. Thank you. Bye-bye.
Have a nice day. Bye-bye.